Thank you for joining I Am Possible, which is India's first future tech meets sustainability podcast. Today, I'm delighted and honored to have with me Mr. Kevin Davies, who's a renowned British science journalist and the executive director of the CRISPR Journal and founding editor of Nature Genetics. He has also authored various books, including Breakthrough, The Race to Find the Breast Cancer Gene, Cracking the Genome, Inside the Race to Unlock Human DNA, The $1,000 Genome, and his most recent release, which is Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. So Kevin, it's a complete honor to have you on the show. Really appreciate you taking time being part of the podcast. Why don't we start with a little introduction, your background, and I saw in your profile that you have Ph.D. in genetics. So what made you choose Nature Genetics and CRISPR Journal?
Yeah. Hello, Eddie. Thank you so much for the invitation to come on your podcast. How did I get into publishing and writing? We could spend an hour talking about that. It's a funny story, I think now, because when I was a student back in England and then doing a PhD, my ambition was to become a world famous rock star scientist. I wanted to become a famous professor at a leading university, quite possibly in America. And for a while, as you progress up the academic tree, I'm keeping pace with my peers and I'm thinking, yeah, this is going well. And then it started to dawn on me during my PhD that, perhaps I wasn't quite as smart or as good at the bench as I thought I was or once imagined I was. And then I landed a research position at MIT for my to begin one of my postdoctoral fellowships. And that didn't go nearly as well as I'd hoped. And so somewhat in desperation, I was thinking, I don't think I'm going to cut it as a molecular biologist, lab researcher. I'm not enjoying putting on the lab coat every day. What else can I do in science? And one day I saw an advertisement in the back of Nature magazine, the famous science journal, to join the editorial staff of Nature. And that was my eureka moment in the laboratory. I thought, hey, that's one way I can get my name published in the world's most famous science journal, by actually joining the staff of the journal rather than trying to submit papers. So I was very fortunate to land that position. That was a little over 30 years ago. And then my big break came a year later when the management decided that the Nature name was too famous, too prestigious to just leave as the title of one journal. Why not try to brand it, as we would now say? And so they were scouting around for a title, a subject to kind of anchor this new Nature journal. And with the Human Genome Project having just got off the ground, that was a big impetus for saying, well, genetics seems to be as hot a field as any. And they looked around the office in London, and I was the token genetics editor for Nature. So I put my hand up and said, you know what, I'd love the chance to run this new journal. And so we kicked that off in 1992. Nature now has literally dozens of journals in every aspect of both the life sciences and the physical sciences. So it's been a good experiment. And I've bounced around and done many things, and you read many of the books that I have published over the last couple of decades. What they had in common until recently was that they were all about advances in DNA sequencing. What can we learn? How can we treat diseases? How can we learn more about ourselves by understanding the complete genome sequence of a human being? And the technology to do that has moved forward by leaps and bounds over the last 2 decades. But I was so fixated on that I really wasn't paying attention when breakthroughs started to be published in the field of gene editing. So I was a little late to the party, but once I learned about CRISPR and what it could do, I was absolutely hooked. And so about 5 years ago, I decided to do 2 things. The only 2 things I can do professionally. One is launch a journal. So we launched the CRISPR Journal in 2018. And 2, write a book. And I wrote a book that I'm very proud of, which you mentioned, called Editing Humanity. And obviously, I look forward to telling you a few stories from that book during our discussion.
Lovely. Kevin, really appreciate you sharing your journey.
Yeah.
So you know what I mean. Before we jump into the DNA sequencing or genetic editing, I want to pull you back a little bit. I want to start because there's a lot of my audience is students. And there are a lot of them who really do not know what a DNA is. So can we start by explaining what
Sure.
DNA is and its role in human biology?
DNA is the genetic material, the book of life. It's the instruction manual that encodes all of the proteins that make up the human body. As a species, human beings are incredibly similar at the genetic level. We are something like 99.9% identical to one another. But as we zoom into the DNA sequence, which is made up of 4 chemical subunits or letters, we just abbreviate them by the first initial, A, C, T, and G. This is the genetic code, and variations in that sequence spell out many of the differences in human biology, differences in our height, in our size, in skin color, and to some degree in terms of behavior and other human traits. So the structure of DNA, of course, was famously solved in Cambridge, England in 1953, the double helix. In fact, we're recording this interview on DNA Day, which is the anniversary of the publication of the double helix. And we've made tremendous strides in completing the sequence of DNA that is bundled in human beings into 23 pairs of chromosomes. And we've learned a lot in the last 2 to 3 decades about mutations in our DNA that cause genetic diseases, and we've identified the genes that are defective in thousands of different genetic diseases. We're learning much more about the gene variations that give rise to more common diseases, diabetes, heart disease, mental illnesses such as schizophrenia. So huge amounts of progress. But what we'd ultimately like to do, of course, is take that knowledge and say, okay, how can we now cure those diseases or treat those diseases? And so we have the pharmaceutical industry and the biotech industry that are devising all kinds of molecules and clever, innovative strategies, including gene therapy, to treat more and more of these diseases. But for me, the reason that there is so much excitement about gene editing is that now, just within the last few years, we have a technology that can actually, and it sounds like science fiction, but now it's science fact. We can actually go into the cell and take this, what the Nobel Committee called CRISPR, the genetic scissors. We can take this scissors or this scalpel and literally make repairs to the human DNA sequence and alter the sequence of a gene, and in some cases fix, literally repair the broken sequence. And I can tell you, as somebody who trained in genetics 35 years ago, I don't think we ever dreamed that this day would become possible. It was so far out there. It would probably have even been dismissed as the plot of a science fiction movie. And yet, in the last couple of years, papers in the New England Journal of Medicine, the world's most famous medical journal, have announced and presented essentially cures for some terrible genetic diseases, including sickle cell disease. And we can talk more about that. Amazing accomplishments. But the catch is this is still a very experimental therapy and a very costly therapy. So how we deliver this to the thousands, if not millions of patients around the world, particularly in less developed nations, is a huge challenge for the future.
These technologies now are opening giving us answers of some of the deepest questions that the mankind has ever known, and we just getting the answers. When you open up the conversation that 99.9% of all human DNA is completely similar, how magical and how mysterious is that? And we need to kind of probe that further, that how has the creator created life? Has it been all copy paste? I mean, he's kept it extremely simple, but that 1% gives us the entire uniqueness of our entire, civilization.
Yeah.
That itself is mind boggling. I'm sorry, you're trying to say something.
No, well, I was going to say, if by the creator you mean evolution, yes, there has been a lot of copying and pasting. I mean, we can see in the evolutionary record as we compare the human sequence to that of chimpanzees and other primates, we can literally see the points in evolutionary history where, for example, two chromosomes have fused to create what is now one of the chromosomes in the human genome. So there is visual evidence. And of course, as we sequence species after species, we see that as you go back further and further in time, the amount of DNA similarity goes down. So we are 99.9% identical to each other, 99% identical at the sequence level to chimpanzees, and then the sequence similarity goes down. But we share genes in common with the banana. So life is an amazing thing. I don't pretend to be an expert on evolution, but there are thousands of genetic diseases that in some cases are caused by the simple misspelling of one letter, one subunit of DNA in a C. The human genome is 6 billion letters of genetic code, and it can only take one typo in one of those letters that leads to the a change in the amino acid sequence of the corresponding protein. Sickle cell is a great example of this, and the results are catastrophic. And so for decades we have been trying to figure out how can we reverse the symptoms of these diseases. And we're seeing great progress.
When I said the creator, yes, I was hinting at evolution itself, because I guess so far there has been so many wrongs. I mean, we've held on to these beliefs or religions and or structures, and which has created so many problems. But I guess science and technology is gonna completely take us into a world which is magical and play a huge role in the evolution of mankind. Before we get into the human editing humanity, I would like to talk about your book, The $1,000 Genome, and when and what do you think it would take for sequencing genome to being a must for healthcare?
Oh, that's a fantastic question. I think so. Let's just quickly step back. What is the $1,000 genome? I had in 2001, I had just published my book about the Human Genome Project, which was called Cracking the Genome. And then in 2001, 2002, I was sitting in a conference, and one of the scientists who was heavily involved in the Human Genome Project, Craig Venter, started talking about we need the $1,000 genome. And I nearly fell off my chair, because the Human Genome Project famously cost about $1 billion. And now here is one of the most respected and famous scientists in that field saying, we've somehow got to slash the price to $1,000. This sounded like crazy talk. But that phrase stuck with me. It took me several years to finally see a path to actually writing a book with that subject. And one of the reasons, two things happened in around about 2007. Firstly, we started to hear of famous scientists having their complete genome sequenced. Jim Watson was one, Craig Venter was another. So I thought that was impressive. Now a human being can have their full genome decoded. Interesting. And then I was introduced to the co-founder of a company that now a very famous company that I'm sure many of your listeners have heard of, and some have even used, called 23andMe. So here was a company in Silicon Valley. One of the other co-founder was at the time married to one of the founders of Google. that's Anne Wojcicki, and I was speaking to Linda Avey, and they were saying we want to democratize the human genome. We want to make genome information available to every man and woman on the street. That company and Ancestry, of course, is another one, has really, it's been fascinating over the last decade. So many people have learned about rare disease genes, rare disease risks that they have in their family. Many have also learned about made family connections. So for people, for example, who've been adopted, this is a way that you can trace to find biological relatives. There is a clinical trial underway in America at the moment, in Boston, called Baby-Seq, in which the investigators are taking a group of newborn babies, sequencing their DNA completely, and then asking over the course of the next 5 to 10 years, is their health improved by having as this resource the full genome directory for those children. There was another story published earlier this year in which a group on the west coast of America, in California, set a new Guinness World Record for the fastest full genome sequence to diagnosis. So when newborns are born and who are born unhealthy and are rushed to the intensive care unit, it's often a race against time to find out what the disease they may have. And one way to doing that is, of course, to read out their complete DNA sequence. But remember, it took us over a decade to sequence the first genome. We can now do this in a matter of hours. This is the new Guinness World Record. And in some cases, then we have a full DNA sequence, and the scientists and the informaticians can then probe that sequence and the computer can ask, where is there a telltale variation that may match the symptoms that this newborn child has? And in a portion of those cases, the match is perfect and treatment can then begin.
In the beginning of the conversation, you were mentioning about CRISPR-Cas9, the genetic editing tool, which is completely upending science. And then you have written a book, Editing Humanity. Would you like to talk about that book, and how has been the reception? Has the Moral Brigade gone bonkers?
The reception has been great. Thank you. I mean, the book was published in hardback. It was published in America in 2020 in October, literally one day before the Nobel Prize was awarded to two of the women I portray in the book, Jennifer Doudna and Emmanuelle Charpentier, who share the Nobel Prize for Chemistry for the discovery of CRISPR, or the development of CRISPR as a gene editing tool. I'm sure everyone in your audience has heard the term CRISPR by now. The book is an ambitious book because I write it in 4 sections, and I try to cover a huge amount of ground. So in the part 1 of the book, I tell the history of the discovery of CRISPR and to share with you stories about the scientists who did this exceptional work. In part 2, I talk about how CRISPR can be used for gene therapy and the rapid progress that is being made, while also talking about the history of gene therapy and the setbacks that the field has had. Then in part 3, and I'm sure we'll talk a little bit more about this in the second half of our interview today, the scandal that erupted around CRISPR and designer babies a few years ago, which I hadn't seen coming when I started writing the book. So that was a major shift in course and direction for the story. And then in the closing chapters of the book, I present some other, just sampling the wide array of applications of CRISPR, from engineering better plants and crops, which is so important in India and places, you have so many people to feed. And without CRISPR, we are going to succumb to pests and bacteria and diseases and drought and floods. And so we need to apply technology to help, or this, we are going to be in very serious trouble. But there are many other exciting applications as well. I'm not going to go into a huge science lesson here, but the fascinating thing about CRISPR is that CRISPR was not designed as a gene editing tool. What the story of CRISPR begins the way that science should begin. It begins with a small group of scientists in Asia, in Japan, in Europe, studying bacteria just because they wanted to understand something about the way bacteria live in certain environments. They had no interest in gene editing. They had no interest in personalized medicine or gene therapy. They just wanted to understand a little tiny bit of biology a bit better. And they discovered around 20 years ago that, that bacteria store in their genome, which is much, much, much smaller than the human genome we've been talking about. They discovered that bacteria carry a strange set of sequences in their bacterial genome. And two European scientists were talking about these funny repeated sequences, and they came up with the acronym CRISPR. That's where the name CRISPR came from. And alternating between these repeat sequences were other short sequences of DNA that just appeared to be complete random sequences. And so the question was, what are these sequences? What is this funny motif in the genome? You talked about copy and paste. Copy and paste doesn't happen really by accident. And a funny structure like this must have some function. That was the speculation. And a European scientist named Francisco Mojica in Spain discovered that some of the sequences within this CRISPR array were actually copied from viruses. And that was sort of the light bulb moment that said, what we have here, bacteria obviously have a way of copying and storing and stitching into their own DNA viral sequences. Why would they do that? The only logical explanation was that this was a type of immune system. And so, the world has been giving us, injecting vaccines to help prime our immune systems to shake off the threat of COVID. Well, bacteria have been doing this for hundreds of millions, if not billions of years, storing viral sequences so that they know if they encounter that virus again, they have now a way to sequence identify the virus, and then they can destroy it. And the way they destroy it is with the enzyme that you just mentioned a few minutes ago called Cas9. So Cas9 it's the scissors of the genetic scissors. But the way it uses it is to activate and express and read out one of these viral signatures that it has stored into a copy of DNA called RNA. And then this tiny RNA segment is kind of bolted on to the Cas enzyme, and that provides the GPS signal that says, go find a match to this sequence. And if you find that match, then bingo, you'll be activated and you can cut the DNA. But a few years later, scientists began to think, well, hang on, if bacteria can take these viral sequences and use it to programmably cut a particular sequence in a virus, what if we put in a different sequence? What if we put in a human sequence or a plant sequence or a mouse sequence? Could we then program this Cas9 enzyme to cut a different DNA molecule? Because a DNA molecule from any species, it's basically the same. It's just a string of A's and T's and C's and G's. It's just the order changes. And so that idea was first floated in 2009. It didn't go anywhere at that time. But then in 2012, the two women we mentioned who won the Nobel Prize, Charpentier and Doudna, they'd met the year before at a conference in Puerto Rico. they decided they would form a collaboration to better understand how CRISPR-Cas9 works at the molecular level. And that was when they published that their work led to the publication of a breakthrough paper in 2012, in which they had simplified the machinery to just the enzyme and one stretch. It's called in the jargon, we call it a single guide RNA that you can sequence in the laboratory to program to cut any DNA sequence. And they realized that they had, at that time, the basis for a fundamental gene editing tool. One of the reasons we're talking about CRISPR today, 10 years after the Dowden and Charpentier paper, is that CRISPR has swept the world. There are scientists in every country in the world using CRISPR because it is so easy to use. You don't need some $1 million piece of American or Chinese instrumentation sitting in your lab. It's just done with little test tubes and pipettes and liquids and stuff that you can order off the internet, little sort of DNA and RNA reagents. And so high school students are doing CRISPR experiments, and it provided this tool that scientists within the space of a couple of years were all around the world eagerly taking up, using their imagination to think of new experiments. And of course, at the same time, major venture capital companies were meeting with Dowden and Charpentier and the other leading scientists in the field and launching biotech companies. And it is those biotech companies that, for the most part, have been pushing the use of CRISPR now into the clinic and leading to some very early days, but very exciting results in treating genetic diseases.
Yeah, let's open up the conversation on the application, because you said 2012, when Jennifer Doudna and Charpentier wrote the paper, it's 10 years down the line.
Yeah.
What's the current advance in the lab today? What's the update with the CRISPR technologies? And share some applications which...
Let's start with the treatment of genetic disease. And of course, Exhibit A is sickle cell disease, which is a very important disease in India and Africa. It is very common in areas where malaria is a major problem, because it's an extraordinary story of evolution. But if you carry one copy of the quote unquote sickle cell gene, you are at an advantage against malaria. You are resistant to malaria. So this keeps the frequency of the sickle cell trait high in these populations. And so that means the chances of 2 sickle cell carriers starting a family high, and of course 1 in 4 chance of them having a child who inherits 2 copies of the sickle cell gene, which means they will have the disease. And it's this terrible disease that, ironically, it's one of the first diseases. It is the first disease we understood at the molecular level, going all the way back to the 1950s, but is one where we have made the least amount of progress in treating, for many reasons, including, I think, racial discrimination. It is not a disease that has been well funded, certainly not in America. And yet we are now seeing many strategies using CRISPR and gene editing to tackle sickle cell, and the results have been spectacular in a handful of patients. So a heavy asterisk here. This only applies to a small number of people, but the results have been tremendous. And in America, there's been a lot of publicity for a woman, an African-American woman named Victoria Gray, who was one of the first volunteers in a trial led by CRISPR Therapeutics. She has sickle cell disease, and so she underwent this experimental treatment. It's a very difficult treatment. It involves chemotherapy. It involves removing stem cells from her bone marrow, then giving the patient a kind of a form of chemotherapy, editing the cells in the lab, not in the body, but in the lab, the cells that you've taken out from the patient, and then reinfusing those edited cells, hoping that they go back to the bone marrow and repopulate in the bone marrow, and now start making the blood cells that have been edited and reprogrammed. And in the case of Victoria Gray and many others in these early trials, the results have been fantastic. And the strategy simply has been, rather than try to fix the broken gene, which is in a gene called beta globin, scientists have known for some time that another way to potentially treat sickle cell disease would be if they could switch back on another form of the globin gene that is active when we are fetuses in the womb, but then is shut down naturally a few days after birth. But the gene is still sitting there in our genome. It is perfectly intact. And if we could just figure out a way to kind of reawaken that gene and switch it back on, the theory was that that gene could compensate for and mask the defective gene. So it's a very clever workaround, and it worked. It worked using CRISPR. And so Victoria Gray, it's now been nearly 3 years since she had that treatment. And from what I have read in news reports, she has been pain-free, no more pain crises that afflict sickle cell patients. They are excruciating. No more blood transfusions, no more hospital visits other than routine checkups. She is healthy. She's been flying around America, and it is a story almost too good to be true. Now, obviously, the physicians managing that trial want to continue to follow her and will follow her for many, many years to come. And of course, at the same time, they are treating other patients, learning from these other patients. And the hope is that eventually in the next few years, that trial will be complete. And then they will go to the FDA and the EMA in Europe and the corresponding organizations in India and other countries to seek approval. So that's the good news. It is breathtakingly good news. Now, the catch is, how much is this therapy going to cost? And I think here, unfortunately, we have some bad news, because the companies that have developed these therapies, these are publicly traded biotech companies with hundreds of employees and very expensive. CRISPR is easy, but the whole process of running any kind of clinical trial is expensive. And so these companies are looking to do right, not only by the patients, but sadly, in this capitalistic society that we work in, they're also looking to do right by their investors and their shareholders. And these two forces are not always in alignment. So that these companies are going to be thinking and calculating what is that middle ground price where I can still make a profit, and hopefully a large profit, so that my investors are happy and then will pour in more money into my company so that I can repeat this process for another disease, and maybe we'll go on and tackle cancer next. But how can I do that while at the same time pricing my new 21st century therapy low enough that there won't be an outcry and the patients will be able to afford this? So, and there have already been gene therapy drugs approved in America with price tags of $2 million. Wow. And this isn't just a problem that faces gene therapy or gene editing. Another company, a biotech company in Boston, Vertex Pharmaceuticals, has over the last 5 to 10 years developed brilliant, successful drugs for cystic fibrosis, a very common genetic disease that mostly affects people of European descent. But I remember a couple of years ago, they could not reach an agreement with the UK National Health Service to get these drugs deployed in Great Britain, because Vertex wanted a high price, and the National Health Service said, "You're holding us to ransom. We can't afford that." They did finally reach agreement, but so this is a challenge, and I hope it's going to be many years. But we've got to figure out a way that we can use the flexibility and the affordability of CRISPR as a raw research tool to help help patients in Africa and across Asia and around the world who there's no way they can afford $2 million. So over the last few years, new flavors of genome editing for example, one is called base editing, another is called prime editing. These have been developed, they are being commercialized in the United States. And these offer the prospect of not just working around a genetic disease problem, but actually now literally the holy grail of literally going into the DNA, into the double helix, looking for that one letter that is misspelled and performing chemistry on the double helix to flip that letter back to what it should be. And this is being applied for sickle cell disease and a handful of other diseases. Again, just starting to make their way into the clinic now. So it will be probably 5 years, maybe more, before these drugs are finally approved. But that really is where I have to sort of pull what little hair I have left out of my head out and say, this is extraordinary. This would have been science fiction 20, 30 years ago. And look what we're on the cusp of doing. It is amazing.
Right. I think there is a convergence of tech, and there are some entrepreneurs who are doing some crazy thing. I think they're pushing the boundary of what could be possible. At least here, what I'm seeing is there's this convergence of things, and these entrepreneurs are pushing the boundaries in the sense, they're defying the odds. Obviously, capitalism will always create a game where it'll be difficult to democratize this and make it accessible for everyone. But I think that the new breed of entrepreneurs are pushing the boundary, and there are biohackers out there trying to see if this could be more accessible to everyone. And I hope that rather than the long 4 or 5 year horizon, I hope that this gets accessible for everyone maybe sooner, because there's so many health problems. There was this talk which I was watching, just it's on YouTube by Bill Gates. The talk is titled "How CRISPR Could Save Lives and End Diseases." That itself is a tall statement. Then there's the Andrew Hessel, the founder of Human Genomics. He says he wants to synthetically engineer life. Then there's this, for the longest time, we have held the belief that aging, disease, and death is an unalterable eventuality. How close are we from altering these assumptions and going beyond?
Oh, you've raised some great examples there. I spent a chapter in the book talking about how CRISPR could revolutionize farming and the production of crops. And I think that is perhaps the most practical arena where CRISPR will really pay dividends. One example I saw, I was watching a film yesterday from The Economist, and I talk about this example in the book, Editing Humanity, and that's about the banana. We all enjoy eating a good, creamy banana, but there are hundreds, if not thousands, of different varieties of banana, and yet we eat only one variety. It's called the Cavendish banana. It has the right properties. It's sweet enough. The husk is strong enough to withstand the trauma of shipping it in crates around the world. And yet this crop is being killed by a disease called TR4, and basically the entire world's banana population is at risk unless we figure out some ingenious way to genetically inoculate or strengthen the banana. And we could make similar stories for all kinds of crops and fruits and vegetables that feed the planet. So CRISPR is a solution where if we understand the pathway that is being attacked by the pest or the parasite or the bacteria or whatever, we can help introduce some resistance to that particular threat. CRISPR, you can rationally do the experiment make the correction that you want, just as you can, when you've written a book, you can do a spell check and you can use your word processor to say, oh my God, I've misspelled Dr. Doudna's name. I must go and fix that before the book is published. It's the true story. Or I've misspelled CRISPR. That also happened in one case in my book. Let me go in and use the word processor to make that little fix. And no one will know that I did that. And that's the beauty of CRISPR. So I think that is one area, and I think most experts agree that agriculture is where CRISPR will have perhaps the biggest influence, certainly in the short term, on human health and longevity. You mentioned biohackers, and I didn't want to really go too far down that road in my book. It's been covered in other places, because I think if you're trying to use CRISPR, it's one thing to order CRISPR off a kit and do some experiments in your basement to show the potential of CRISPR. It's another to start injecting yourself thinking that you're gonna, I don't know, put on 5 pounds of muscle or whatever. That is a little foolish. It's a little silly. If anything, if there was to be any kind of mishap, you'd only have yourself to blame. And so I don't think we want to be encouraging people to go down that road. I think from the medical perspective, this has to be done responsibly and ethically. And so I'm glad that is happening. At the end of the book, I talk about some of the fun examples, and I'm sure you can almost devote entire episodes to these. There's the thoughts that perhaps we could resurrect the woolly mammoth and start seeing herds of woolly mammoths roaming Siberia again to keep the frozen tundra packed down, to stop all of the megatons of methane and carbon dioxide from leaching up into the atmosphere. And on the CRISPR Journal, we've just published a very fun paper. It's been our most, the paper in the 5 years we've been publishing that's received by far the most media attention. And it's in which scientists in America have made progress towards developing a hypoallergenic cat. So the title on the cover of our journal was "Here, CRISPR Kitty?" ? They've identified the gene that causes the cats causes people to suffer an allergic response because it's a reaction to this protein that is shed. And so the scientists have identified the gene that encodes this protein. And the thought is that maybe with CRISPR, in the future, we could find ways to remove that gene or lower its expression to prevent people. It's a very common, at least in Europe and the US, it's 1 in 6 or 1 in 7 people suffer really miserable cat allergies. They can't be in the same room as a cat. And so, maybe not a life-threatening. Well, in some very rare cases, it can be life-threatening. So, there's so many areas where CRISPR is being applied. And so I think, there's going to be more and more CRISPR books in the decades to come, hopefully.
Lovely. Yes, and I would like to, highlight George Church and his efforts over here, because I guess, we need these bold entrepreneurs who have a vision and who kind of go beyond, and that would be so cool, if we kind of do that, bring back, revive a mammoth. You mentioned about shortcuts. Yang Kui, he made headlines, when the genetic editing happened. We are past that, we definitely past that. But would you like to kind of address the moral and ethical implication? And I would like to make this a little clear to my audience, because, whenever we have these conversations of possibly like a technology, there is the larger conversation is technology good or technology bad?
Yes.
I would like to highlight that it's not the case at all. Technology is something that we leverage. It's the humans who choose to use it in a specific way is the output. So if we choose wisely, we'll always have a great future. So would you like to address the moral and ethical implications of maybe bio warfare? or militarizing CRISPR technologies, because there's talks, or maybe I just read article about, creating super strong military, where you could suppress the military force with, of feeling pain and stuff like that.
Yeah.
Does that kind of bother you?
So now human beings are playing God. They are changing the gene pool, one embryo at a time. No responsible scientist felt that this experiment should be attempted. Not now, not with CRISPR being so young. But He Jiankui didn't listen to anybody else. He consulted a very small circle of friends and colleagues and acquaintances, but he didn't share his plans in public. Had he done so, he would have been told in no uncertain terms, "You're nuts, do not do this. The technology is not ready to attempt this." But he didn't listen to those voices. Instead, he dreamed of becoming famous by recruiting couples in China where one of the parents has HIV. And he felt that by using gene editing, he could deliver babies who are immune to HIV by disrupting one particular gene, the gene that codes for the HIV receptor in our cells. And thus he would be celebrated as this superstar scientist who had shown us the path to eradicating HIV. I think that was his dream. It was naive, it was silly, it was scandalous. But he recruited those couples and he went ahead with the experiment. And then, of course, in late 2018, we learned about what he had done. And He Jiankui was vilified almost from the moment that story came out, because he had no right to do this, and he had no right to interfere with the genetic sequence of the two twins who were born in late 2018. So what happened? He presented his work. He was condemned by the international community. Later, he went to jail. The Chinese authorities realized that he had gone too far, and he was eventually taken to court, and he has just been released after serving about 75% of his 3-year prison sentence. He is not allowed to work in this field again, and I don't know what will become of him. There have been commissions that have studied what he did and tried to assess, to your question, what is the future of human embryo gene editing? Are there any circumstances under which this could potentially be justified? And the major committee that looked at this, with some brilliant scientists on this panel, said yes, there are circumstances where we think this might be viable in the future, where no other fertility treatment is available to a couple. So if parents both with sickle cell disease decided they would like to have a healthy biological child, they have no other ways to do this. They can adopt, of course, but that's not a biological child. Because they cannot use any fertility measures because mum carries 2 copies of the sickle cell gene, dad carries 2 copies of the sickle cell gene. There are no healthy counterpart genes to select for using IVF. Gene editing could potentially be a solution to them. So we will have to see where this goes in the future. You talked about biowarfare, which of course is another application. I'm not worried about CRISPR being used in biowarfare because human beings have already devised so many terrible technologies that can wipe us off the planet. I mean, there is the threat of nuclear power being deployed in Europe right now. That's a very real threat. So I'm not really worried about, and we've seen the threat of a virus. There's still some debate, I suppose, about whether it was leaked or possibly even conceived in a virus lab in China. I don't believe it was man-made. I believe it occurred naturally. But be that as it may, we've seen the havoc and the death that nature can level on us. And we're seeing it every day, from climate change and the floods and extreme weather and the fires. So worrying about somebody in a lab, that you don't need CRISPR to develop a very nasty weapon. So that is not something I spent too much time thinking or worrying about.
In the beginning of the conversation, you mentioned that you wanted to be a rockstar scientist, and there are rockstar scientists who are doing some really, really cool thing. And one of them who I really look up to and who's doing crazy thing according to me is Dr. Michael Levin from the Tufts Lab. He's doing some seriously crazy thing. Who, according to you, are the rockstar scientists? And paint a picture of a world. What the world is going to look like in the next 10 years, when the genetic editing CRISPR technology is going to be accessible for everyone.
He would shudder if he hears this, and probably wouldn't want to be described as a rock star. But the scientist who I think has most captured my imagination during the writing of this book is David Liu, who is a chemist at Harvard University and the Broad Institute in the Boston area. It is within his lab that these two new gene editing technologies, base and prime editing, have been developed. So David's forte is he recruits brilliant young scientists to join his lab, either as graduate students or postdocs. And together they work collaboratively to devise these new chemistries. And so he is literally the leading the field in performing chemistry on the human genome. And this is where I think what was a complete fiction 30 years ago has become science fact. So, I could see somebody in that field potentially winning another Nobel Prize, this time for medicine, if the potential of these new precision gene editing technologies comes to fruition in the clinic. So I think that's amazingly exciting. We have to be patient. CRISPR is in many ways a miraculous new technology. But as I tell the story in the book, it came about because a handful of scientists just were fascinated by why bacteria could survive in weird environments on the planet, volcanic subthermal vents and high salt conditions in various lagoons and places. What was it about their genetic architecture that allowed them to thrive in these very hostile environments? So from studying fundamental questions of basic biology, we've now, from that, has sprung CRISPR as we now know it and as is celebrated in films and books, this gene editing technology. Who knows where the next technology will come from? There's lots and lots of new companies being founded that some say we can change a single base sequence. Others are saying this is the technology where we can now make bigger changes, more widespread changes in the genome. So the playbook, the toolbox for manipulating DNA is evolving and is improving all the time. And I think what we've learned is not just simply, let's not speculate where things will be in 10 or 20 years, because chances are we'll have moved even further ahead than we even dare to predict. So that's my prediction. Well, no, Eddie, I just wanted to thank you, and I hope people listening will check out Editing Humanity. You may have to ship the book from overseas, but it's hopefully well worth your trouble. And I also narrate the audiobook. So if you've enjoyed this interview, you might enjoy the full audiobook of Editing Humanity.
Thank you, Kevin. Really appreciate you taking time, being part of the podcast, sharing your insights. Yes, we in a fantastic point of time with this genetic editing CRISPR technologies, and I hope that there is more entrepreneurs, more researchers, more scientists join their hands to making this accessible, because unless and until it's not accessible to everyone, I mean, it'll just stay for those really rich people. And I hope that this upends healthcare, and it's all that this technology has got the possibility to do that. So thank you. Really appreciate you taking time being part of the podcast, and to my listeners. Lovely. Obviously, I'm going to leave the link behind also. Kevin, really appreciate it. Really appreciate it. Thank you.
Thank you, everybody. Thanks, everybody.
Yeah.
Bye-bye.
Bye. See ya.